Welcome to today's Student Journal Club and Networking Program, hosted under the Bioanalytics Skill Development Forum. We are honored to have Dr. Manisha Choudhury as our distinguished speaker. Dr. Choudhury completed her PhD at the Center of Advanced Studies in Crystallography and Biophysics at the University of Madras. She then served as a visiting fellow at the National University of Singapore and pursued her postdoctoral studies at IIT Bombay. Today, Dr. Choudhury will be sharing her expertise on snake venomics, unraveling the complexity of venoms. Please join me in giving a warm welcome to Dr. Manisha Choudhury.
Just to give my introduction, I'm Dr. Manisha Choudhury, and I have worked as a postdoctoral fellow in IIT Bombay from 2019 to 21, and currently I'm working as a postdoctoral fellow in Department of Zoology, GAUHATI University. So today we will discuss about snake venomics and this field of research, and wherein we will talk about unraveling of the complexity of snake venoms. Next slide, please. Okay, so why should we understand snake venoms, or why should we work on snake venoms and be interested in this field of research? So basically, snake venom, it is a complex mixture of enzymes, which are proteins and peptides, and also several kinds of other compounds. And most of it, these enzymes, they are responsible for giving the property of toxicity to these snake venom, and also certain protection or else defense purposes for the snake. So as you can see that these enzymes, they possess several kinds of bioactive potential, such as they might be they might serve as antivirals or anticoagulant or antiplatelet activities. There are several research papers which are already available, which talks about the anti-cancerous property of these proteins and peptides. And further moving on, so this is the discussion about the pharmacological potential of these enzymatic proteins and peptides. However, also because we know that snake bite causes death, right? So which means that these snake venoms containing so proteins and polypeptides, they will definitely have their toxicological effects. So it is another aspect wherein we also have to study about the cytotoxicity, neurotoxicity, and also the myotoxicity of this particular proteins and peptides. Before going ahead with the pharmacological potential, we also have to look forward that what are the side effects that uh, can be caused by this particular proteins and peptides. Now, already, and this field is very interesting because already we have drugs in the market wherein these snake venom proteins and peptides, they have been used as the basis for designing pharmacological drugs. For example, here the first one is captopril. So captopril, it is actually designed based on the bradykinin potentiating peptide, which was isolated from particular Bothrops zararaca venom. So Bothrops zararaca venom, so from there they have extracted bradykinin potentiating peptide, and this has uh, built the foundation for designing captopril. And captopril is already available in the market for treatment of cardiovascular diseases. So similarly, there are other several drugs wherein you can see the most significant ones are the slide. Okay, so another one is the hemaggluteinase. So that is also another one which is very significantly, it plays a role in coagulation. So maybe we can move to the next slide. So now, so there was this interesting question from one of our guests here. So that how can we obtain snake venom? Because this is one very important question. Uh, wherein suppose you're working in Europe or any of the other countries outside India, here, let us talk about Singapore, or we can also talk about Australia. So extensive research work has been going on snake venom, and there we have this provision of keeping the snake in captivity with authorized permission. So these research institutes, they have their authorized permission from the forest department, so they can keep the snake, they can go and catch the snake and keep the snake in captivity, milk the snake. First of all, you have to feed the snake, then you can milk the venom, and then further take that venom for your research purposes. And then you have to release the snake in the uh, proper condition, the way you in which you bought it from the jungle. Then you have to go and leave it in its own habitat. So that is what we did in National University of Singapore while I was doing my snake venom research work. So we went to Bukit Timah Nature Reserve in Singapore. So we collected, so this is an acutus. So this particular snake, so we caught it, we kept it in our captivity. We fed it, and then we milked the snake, and then we used the venom for our research. But then, next slide, please. In India, we have this issue that we do not get permission like this. So we cannot go to the jungle, catch a snake, and keep it in our research institute in captivity and milk it. So it's not possible. So here, what we can do is, there is a tribe. Okay, many of you might who are from South India, you might know about the Irula tribe. So these people are very, very experienced snake catchers. So and this is a tribe which has been striving on like when there was this we India used to export snake skin. So now it is banned. So when India used to export that, so that was a business, there was a business of snake skin. So this people, so this Irula tribe, so they used to thrive on that. Their bread and butter was on that. So then after that, what happened? They, although they had this, unique potential, unique capability, but then there was no much, I would say, focus, or I would say, uh, they were not known across the world or even across India. So there is this person in this group. You can see Romulus Whitaker. So this particular person, so he's a citizen of USA. So he came to India when he was only 10 years old, 10 or 12 years old, with his father, and he visited the Irula Park. That Then he actually was so interested, when he could see the Ophiophagus hana, that is the king cobra. So he really loves king cobra, and so he stayed back. He actually founded this particular Irula Snake Catchers Cooperative Society, and which now is actually India's biggest producer of antivenom. So, and this particular person, Romulus Whitaker, he is also the founder of India's first snake park, which is in Chennai. So he helped this particular tribe, and now this particular Irula Snake Catcher Society, they produce their own anti-venom. So how? So first because they are good snake catchers, they are authorized. They will catch the snake, they milk the snake venom, and then they sell it commercially. So if you want to purchase snake venom for your research work, then you have to get your permission from your state authorized uh, your institute, and then finally, and you have to just give a brief summary of the research proposal, how you are planning. Then you can send it to them, and also they have their own forms and everything, documents to be filled up. After that, they will check with their if everything is verified, then you can purchase the snake venom from them. So this is the only method wherein we can purchase snake venom in India. Please go to the next slide. And one more thing I would like to say, the good thing is that already from this Irula snake tribe, 2 persons have been awarded Padma Shri in 2023, if I'm not wrong. So now they have also got their recognition, and it will be good if we plan to work, so we can contact the Irula Snake Cooperative Society. Now, just to give a brief overview of snake venom and the snake venom proteins. So, serpent is the group, okay, that is the snakes group, the superfamily. It consists of around 16 to 20 families, and around 2,800 snake species are known till date. And all the venomous snakes, they mostly belong to Colibri family. So envenomation following snake bite, so it causes considerable morbidity and mortality, and however, it still remains a neglected threat to public health. So there are records wherein it says that almost 1.8 million incidences of snake bite occur worldwide, and which leads to approximately 94,000 deaths. So till date, antivenom is the only effective treatment for snake bite. However, we will also discuss what are the disadvantages that are faced till even today. And so here we can see that the first disadvantage I would say is if you can have a look at the particular snake venom antiserum, below this it is written as polyvalent. So polyvalent means the antibodies are not for only one kind of snake. So in India, the major four species of snakes which are causing huge number of death are Indian cobra, Indian crate, Russell's viper, and saw-scale viper. So these are called as the big four. These big four are actually injected into the particular organism for extracting the antibodies and making this particular antiserum. So now, but the problem is that these polyvalent antivenoms, they have their own side effects. The side effects are more in case of polyvalent antivenoms, and it is lesser in case of monovalent antivenoms. But designing monovalent antivenoms is another challenge, because when the snake bites, the main issue is we do not know snake has bitten us, right? So when we go to the hospital, so on what basis the doctor would inject us the antivenom? So that is a major problem that we are facing. So here you can see that this image is of a monovalent antivenom for the brown snake. So monovalent antivenoms are available, which has lesser side effects, but then we still in India, we are preferring polyvalent antivenoms. Please go to the next slide. So venom composition. So these are the proteins and peptides which are present in snake venom. So please go to the next slide. Okay, so now, because the first and foremost thing, if we're talking of designing antivenoms or identifying new proteins and peptides from snake venom, so we have to understand the composition of the venom. So to understand the composition of the venom, so there are two kinds of approaches. First is we can go for transcriptomics studies, and the second is we can go for proteomics studies. But the issue with transcriptomic studies is that for transcriptomic studies, we will need tissue. And tissue we have to extract from the venom gland. And for that, we have to kill the snake. So this is a very big issue in case of studying transcriptomics of snakes. And therefore, more prevalent is the proteomic studies. So because in case of proteomic studies, if we want to carry out proteomic studies, then we can actually get access to the venom of the particular snake species, and then we can go for a mass spectrometry analysis studies, and then we can identify might be novel peptides, or in fact we can also get an idea about the composition of the venom. And of course, it is also very crucial to map the combine transcriptomic studies along with proteomic studies, because we know that Proteomics can only provide the information about the post-translational processes. So, the information about which we might miss in case of transcriptomics. Next slide. Okay, so here we will just, I'll just take you through a case study. So wherein I'll show that, how we can actually get an understanding about the composition of the venom. So in India, there is estimated that 35,000 to 50,000 human deaths are caused due to snake bite. And majority of the bites, as I have already mentioned, is caused by the big four, that is Indian cobra, common krait, sawscale viper, and Russell's viper. And so it is very important that we have to understand the composition and overall biochemistry of individual toxins. And so here this is about how we can evaluate the composition of venoms of two elapids, which is one is the Indian cobra and another is the common krait. This particular field of research, wherein we understand the mechanism of venom toxicity and analyze the venom composition. So we have to characterize individual toxins. That is also very important. So each of the individual toxins that I have mentioned already, that there is this entire list of proteins and peptides which have been identified. So each of these proteins and peptides which are present in snake venom, and which also there is a high chance that we might come across several novel peptides, because one thing I would like to mention here is the snake venom composition differs based on geography, differs based on their food habits. So if I'm talking about Indian cobra in South India, and the venom composition, it will very much differ from the ones which I find in North first, and it has been proved by scientific evidence. That is why we cannot say that because Indian cobra is characterized and then there is no further need to study geographical composition. Next slide, please. Okay. So this is a case study. Wherein we have done proteomic studies for the Indian cobra, that is the Naja Naja, and Indian krait, that is the Bungarus caeruleus. So we have used three approaches here, because in proteomics, in case of particularly snake venom, so we do not have a necessary very I would say very defined protocol which we can use for any kind of proteomics characterization. So we have tried our best to compile three methods so that more number of proteins and peptides can be identified. So first, in the first approach, I have done in-gel trypsin digestion. Then second, in solution trypsin digestion, I have combined all the three, and then I have used. So individually, I have done tandem mass spectrometry for each of these samples. And then finally, we have detected the proteins and peptides. And then also we have designed a script wherein we try to analyze how these proteins and peptides are mapping to the actual sequence. Next slide, please. Okay, so in the first approach, what we did was it's very simple. So we have just run a 12% polyacrylamide SDS page, and I have loaded 20 microgram each of the crude venom samples in the lanes. Then you can see how the pattern, the bending pattern is changing for Indian krait and Indian cobra. So the lane 2 is Indian cobra and lane 3 is Indian krait. So you can see how the bending pattern changes. Although both are elapids, but still the bending pattern changes. So the lowest molecular weight are for the three-finger toxins and phospholipase A2. Then we have venom nerve growth factors and cysteine-rich secretory proteins. We also have then further we have snake venom metalloproteinases, L-amino acid oxidases, and beyond 100 kilodalton, they are the aminopeptidases. So then what we did was we have extracted the slice each of these gel bands into four fractions based on their sizes, as you can see from the boxes. And then I have subjected it to in-gel trypsin digestion and then run on mass spec. So from this, I have identified 25 total proteins in case of Naja venom and 23 proteins in case of Fungus ceruleus venom. That is the Indian grade. Next slide, please. Then in the second approach, what I did was I have subjected the crude venom itself to mass spectrometry. So the crude venom, it was subjected to tryptic digestion and analysis using tandem mass spectrometry. From that, this approach, I have identified 40 proteins belonging to 32 toxin families and 43 proteins belonging to 8 toxin families. And in the third approach, I have run gel filtration and then followed by tandem mass spectrometry of the separated fractions. So here you can see that when I have run the gel filtration of Naja, that is the Indian cobra, it has yielded 9 distinct protein peaks, and each distinct protein peak, each fraction, they were supposed to again tandem mass spectrometry. So I have identified 75 total proteins belonging to 14 toxin families from this approach. And further from the Fungus ceruleus venom, so I have identified 34 total proteins belonging to 8 toxin families. Next slide, please. So now coming to the relative abundance of venom, Naja venom, so that higher percentage of so I would have the image for vipers, then you would have seen that this changes. For example, the percentage of snake venom metalloproteinases in case of elapids and in case of particular viperids, so they differ. Here also you can see that particular ACHE, that is acetalcholine esterases, these are present in case of Fungus ceruleus venom, but they are not present in case of Naja venom. So this kind of compositional variation occurs, and this is very important because for in the future goal, the main goal which we have is of designing monovalent antivenom. So if at all I want to design a monovalent antivenom, it is very crucial that I know the exact composition of each particular species of snake. So here you can see that I have characterized Naja venom. Then I have also characterized, done a calculation of the protein percentages in case of Fungus ceruleus venom, and then I can compare the two, that which kind of particular protein, or which are the 2 or 3 proteins that are majorly present across that particular species, but is absent in the other species. Next slide, please. Slide, please.
You're inaudible.
Sorry. Hello.
Yes, now you are.
Yeah. Okay. Please. Next slide. Okay, so further moving on, we have then gone for the MS visualization program. We have written the script for this. So wherein the output files that we have generated from Sequest and Mascot. So these are the output files. So I have used a Sequest and Mascot softwares. So I have tried to analyze the lysine and arginine rich medium and pore regions, the glycosylation sites, and the peptide coverage. Why? Because the because as you have seen from the experiment that I have used trypsin in most of the cases, right? So trypsin is the major proteinase that I have used for cleaving the proteins and peptides. Now, trypsin actually cleaves at lysine and arginine regions, right? It cleaves at directly after a lysine or a arginine residue. Therefore, if the particular region is very high or low in presence of lysine or arginine, then it has a tendency to be ionized and not be detected in the mass spectrometry. That is, we might miss the particular peptide. So, and also in case of glycosylation sites also, they are very crucial because these cannot be detected. We cannot detect that particular region in mass spec because the mass of the sugar moiety is not predictable. Therefore, it is very important that we exactly predict that why have we lost that particular peptide, and if that particular peptide is very crucial, then do we have a chance of losing it? Right? So this information has to be known beforehand so that we can uh, adjust the settings in such a way that, or we can also change the trypsin. We can change trypsin to chymotrypsin, or we can adjust the experiment in that particular manner so that we do not miss the peptides. Next slide, please. Now you can see here that this particular region, the blue region, that is lysine and arginine core region. The cyan region shows that it has a glycosylation site in that particular position. Then detects, red is showing that this is a lysine arginine rich region. And these small peptides, the small red color dashes that you see, so these are the peptides. And the entire longer sequence that you see is the protein sequence. So this image is actually showing you that these peptides have been detected by mass spec in case of my experiment. So these peptides, I know that these peptides are present in my sample. I'm just mapping it to the particular sequence and checking how these particular peptides which have been detected are mapping to the protein sequence. And also the red region, red bars, they are denoting that these are low scoring peptides. The purple bar shows that these are medium scoring peptides, and the green bars here in the left hand side, you can see the green bars. They are high scoring peptides. Means this is nothing but the accuracy. So how reliable are these peptides? So these kind of images, so we wanted to generate for our manuscript. So we have written this in Perl, and then this is how we have shown that how these particular peptides are mapping. And you can see, as long I have not shown, most probably I have missed that slide. So this, because these are short sequences, you can see more overlaps. But as the sequence gets longer, the overlaps will be lesser. So less number of peptides will overlap in the protein sequence. So that is the reason that we can detect higher number of three finger toxins because they are very short. They are only 65 amino acids or so. But snake venom metalloproteinases, they are very long. So I think around 200 to 300 amino acids. So then the detection gets lesser. So these kind of difficulties we also face in case of, I would say, toxicovenomics. Next slide, please. Now, so that approach we discussed for the proteomics characterization, because if we move on in that approach, then we will get information about what are the novel peptides present in the particular sample. But now, as I said, it is also important that each protein. So as I said, we will detect phospholipase A2, three-finger toxins, venom metalloproteinases. But it is also important that if we find a novel toxin, then we also have to characterize that particular toxin, right? So here in this study, so that study was the previous case study was on Naja Naja, Indian cobra, and Indian grey. So this case study is on the viper venom. So that is the Echis carinatus is the saw-scaled viper, highly present in Indian subcontinent. So the viper bites, they usually result in platelet aggregation, and blood coagulation. So they are very known prominent procoagulants. Then viper envenomation, it leads to local manifestations they call hemorrhage, myotoxicity, that is the extracellular matrix degradation at the envenomated site. So wherein the snake bites, so you can observe hemorrhage and myotoxicity. Then the most prevalent metalloproteinases are the snake venom metalloproteinases in case of vipers, which are responsible for causing pathological effects. And particularly the venom researchers, they have given special attention for the identification and characterization of snake venom metalloproteinases. The reason being that they are very high pro or anti-thrombotic or coagulants. Okay, so snake venom metalloproteinases, they can serve as anticoagulant or they can also be procoagulant. So definitely, so these informations need to be reported, and so the characterization of any novel toxin is very crucial. Right, please. So first of all, if I have to characterize the particular snake venom protein, then I'll have to extract that protein. So I need that protein in the purified form. So there can be two methods. One is that if I can purify the protein, if the protein is present in sufficient quantity in the venom, then I can extract it by purifying it through chromatography techniques. But if the particular protein is present in very less quantity, okay, and I am not able to, even if I'm running chromatography once, twice, thrice, I'm not getting enough of the protein concentration to perform my experiments, then uh, then I have to go for then I have to go for approaches such as cloning and expression. Okay, so I have to do cloning of the particular toxin, or and then further I have to go for expression, and then I can yield sufficient amount of protein for my experiments. So these two approaches can be taken. So here in SVMP, particular ECP3 was present in sufficient quantity, so we prefer to perform the uh, chromatography approach. That have run cytotoxic chromatography. First of all, I have protein to purify the particular protein, and this has resulted in several peaks. Now, because the other thing was that I can go for mass spectrometry of each of these particular fractions for detection of the peptides for this particular ECP3, right? But we prefer to go for a functional based identification. So what we did was, for each of these peak, we went for casinolytic activity assay. I'll just tell you in the next slide what is casinolytic activity assay. So we did casinolytic activity assay, and then so for the peaks which were showing casinolytic activity, so those peaks again we took and then we ran it on an ion exchange chromatography. Next slide, please. So you can see QF4, that particular peak with asterisk. So that peak contains the pure protein, that is the ECP3, that particular fraction. So after doing all of this, so because I have also already mentioned that so I have beforehand I have done the proteomics characterization of this particular ECP3. So I have run this. I already had the peptides for this particular protein detected using mass spec. So here this is an image which shows that the entire protein sequence. So you can see these are all snake venom metalloproteinases with the accession IDs, and the particular disk study, so that is the ECP3 protein. So the brown colored dashes, they are the brown colored dashes, and red, black, and cyan dashes. Sorry, not brown. That is red color. So the red color one is the disintegrin-like domain. So snake venom metalloproteinases, they are divided into different domains. The protein contains different domains, which each of these domains are responsible for different functions. So that's very interesting, because the disintegrin-like domain, the cysteine-rich domain, and so each of these domain, they serve their different function, and Herein you can see that the red color ones, F1, F2, so these F1, F2 are nothing but the fragments. Fragments means these are the peptides that have been detected in our sample. Okay, so F1 to F5, they are in the metalloproteinase domain. F6 and F7, they are in the disintegrin-like domain, and F8 and F9 are in the cysteine-rich domain. Okay, so we have just shown how the particular peptides that we have detected in the sample, they are overlapping with the protein sequences in the database. Next slide, please. Now, okay, so the next approach, if you remember in the second slide itself, I have mentioned that we will talk about the pharmacological effects, that is antiparasitic, anticoagulant, procoagulant, etc., etc. But at the same time, we also have to understand about the toxicological effects, because each of these proteins will have their toxic effects, right? So we also, because ECP3 is now a new protein that we are characterizing, so we have to know that what are the toxic effects that this particular protein can cause. So the first assay that we did was casinolytic activity assay, which I also showed in the particular size exclusion chromatography slide. So what we do in the casinolytic activity is nothing but, you know that casein is a protein, yes. So we use that particular protein, and then we add purified toxin, that is, in this case, this is the ECP3. The positive control will be the crude venom. Okay, so because we know crude venom means the ECP3 venom itself that we have purchased. Okay, the ECP3 venom is the crude venom, and we know that it contains high amount of metalloproteinase, and you know that metalloproteinase means nothing but they will break the protein, they will denature the proteins, right? So that serves, ECP3 crude venom serves as a positive control, and then particular our toxin, it is the test, and saline will serve as the negative control. So you can see in case of casinolytic activity of ECP3, if so this is the OD. So first we will just, so the experiment, actually we will add the casein along with saline, and then another tube you will be adding crude venom, another tube you will be adding the test, that is a ECP3, and then you will check the OD. Okay, so once you in a spectrophotometer, and then you will plot the graph, and you will know that how whether there is positive result or negative result. So here you can see that clearly that casinolytic activity is observed in case of crude venom and in case of ECP3. Okay, so in case of this, the next slide, the next figure, you can see that this is a study on animal model. Okay, so we have done a subcutaneous injection of the particular crude venom, that is the saw scale viper crude venom. 4 microgram was injected in mice. Okay, and then we have taken out the slices of the skin, and we have checked for hemorrhage. Okay, the first one is saline, so you can see there is no hemorrhage at all. The second one is of the injected crude venom. Okay, so clearly you can see that there is hemorrhage. Okay, and but the ECP3, till we have injected 20 microgram of purified ECP3, it did not show any kind of hemorrhage. Okay, so we can see that this particular ECP3, it is non-hemorrhagic in nature, it doesn't cause hemorrhage. Okay, next slide please. Then further, what myotoxicity. So here you can see saline, crude venom, and ECP3 in the figure for myotoxicity for ECP3. So we have first, so CK for calculation of myotoxicity studies. So we usually concentrate on the amount of creatinine kinase that is released after injection of the venom or the purified protein. Okay, so after injection of 4 microgram of crude venom, so we have seen that the activity observed is 5.9. Okay, and then in case of particular, when 20 microgram was injected in case of ECP3, So the effect is negligible. You can see it is just up till the level of saline. Okay, but the crude venom if activity is so high, right? So we can say that ECP3 is non-myotoxic in nature. It doesn't show myotoxicity. Then going ahead, I have already mentioned that vipers are procoagulant. 90% of the vipers are procoagulant. Their venoms are procoagulant, wherein the elapids, that is the Indian crate, the Indian cobra, they are anticoagulant. Okay, so uh, here you can see that particular. So how we have analyzed that? So ECP3. So we have performed the coagulation assay for that to understand the procoagulant activity. So we have noted the recalcification time. Okay. So and how it changes from the normal clotting time, the recalcification time. So the normal clotting time is 175 seconds. The normal blood clots at 175 seconds. Okay. So in case of plasma, you can see that normal blood clot. But when you are adding uh, around zero point 0.5 microgram of the crude venom to plasma, you can see how drastically the change is observed in clotting time, right? Almost 9 times. Yes. And when we are adding plasma with around 5 microgram of ECP3, so it is the clotting time is reduced by half, which means ECP3 is a procoagulant protein. And you can ask me that how we decide on the doses, right? What amount you are saying 20 microgram for ECP3 and all. So how do you decide on the doses? So what we do is these experiments are all dose dependent. So we keep on increasing the dose to an amount. After that, so 20 microgram of the protein is high. Okay, is very high. Suppose this toxin or this protein ECP3 has to be a drug. So then we cannot go beyond 20 microgram anyway, right? So because it's a very high dose. So if I want to design this ECP3 into a drug, so going beyond 20 microgram uh, is uh, is just a waste of time. It will never anyway go beyond for a drug. It cannot go beyond that dose. Okay, so it has to be least dose. So I think around if the activity is not shown till around 15 microgram itself, we should supposedly stop. Okay, so the next slide, please. Now, so coming to the concluding part of my talk. So, because just to make this also a informative talk, so that it relates to our day-to-day life, we might come across cases of venomous snake bite. Yes. So the first thing that we should always do, most of the cases, it is said that you should bind wherever the snake has bitten. You should bind that in tightly so that the blood doesn't flow. Okay, so it's a very wrong concept. Okay, first of all, so here it's very interesting. You can see that keep calm, okay, and note the snake's color, size, and shape. Okay, and you should not get scared. You should note all this if possible. Fine. So I don't think it's possible, first of all, and that is the biggest issue that we face, because once a snake bites, and you will, of course, you'll be in a state of panic, and there's no time to record which snake has bitten you. So when you go to the doctor, so now that the doctor doesn't know what kind of antivenom he should provide, right? He should inject. So they give you polyvalent antivenom. However, you might have come across several news articles also that injecting several times, suppose first time the snake bite, the person is bitten by a snake and he's injected polyvalent antivenom. Again, the second time also same thing happens, and the third time it will be, number one, it will be either the efficacy will not be there, the antivenom will not work at all, or the person will die just because of the side effects. Okay, so this is cross-linking of antibodies. There are so many issues. So this kind of issues will be faced, and then the polyvalent antivenoms finally, uh, that is the issue that we face in India. Okay, so that is why also there was this another very recent incident wherein the person knew, okay, he has gone to many of these workshops wherein they treat how to take care of a snake bitten person. So he has actually literally taken holded that snake, and then he had gone to the doctor to show that this snake has bitten me, and you should please look at this, and then you inject the antivenom. What kind of antivenom you should give me? Okay, so that is the issue. And then the second thing is that never ever put any kind of tight bandage around the snake area where there is the wound. Okay, because that actually causes severe swelling, and there is more harm to that particular wound, and later on many of the cases you have seen that the leg needs to be cut and all, right? So that is the issue. So never, always leave it just like that. And uh, also that region should be immobilized, but not that particular wound should not be uh, very tightly bound. Then after that, you should wash that particular region with clean water, and then just go to the doctor. So thank you, and thank you so much. So I'm open to questions now. Thank you.
Let us say like a time dependent structural analysis or any of those kind of things.
For the software?
No, structural biology studies like protein interaction.
Structural biology.
Yeah,
Structural biology. Yeah. For any of these novel toxins?
yeah, and also
Okay.
the high molecular weight proteins.
Yes. So no, I have not gone for any of the crystallography determination or any of NMR kind of studies for these particular proteins. Yes, because I had to right now, because this was my PhD work long back. So still it is yes, I would say, because ECP3 still it is uncharacterized structurally. So only in silico studies, few of the in silico studies just for modeling purposes and all has been done and deposited. But the structural aspects have not been looked into yet. Few of the proteins we have using in silico approaches, but as in when you talk about structural biology like X-ray crystallography and trying to look into the mechanism, no, I have not gone into that field yet.
So, do you think is there a possibility to do some, I mean, or propose such a project, especially for time dissolved cryo-electron microscopy, or you know, because I think this is important when it comes to VENOMICS, bridging the gap between.
For so if I'm not wrong, you are saying that it should be done with cryo-EM, so it will be
Yeah,
interesting.
it is possible. That's what I'm trying. Yeah.
Yes, 100% it is possible. Yes, it is possible, and it should be done. It is just that I have not done yet because yeah, there are certain limitations and issues, because when I'm in a postdoc, I have to look into the facilities that are in and around me. So yeah.
So
Yeah,
because we are a
definitely.
startup,
This is something I'll
we
be
are a
very
startup.
interested to. Yeah.
Yeah,
Sorry.
because we are a startup trying to bring in cost efficient and also time efficient cryo-electron microscopy approach in India. So from this perspective is what I'm asking, and I would love to have a probably, I mean, initiate a email conversation or a couple of details. Is that okay?
Yes, sure, definitely. I'll
Right.
be very
Thanks a lot.
interested.
Thanks a lot.
Thank you. Thank you.
Ma'am, I'm pretty interested in cancer research. I'm actually just doing my UG though. So in snake venomics, like how can we separate or identify cancer inhibiting agents?
Okay. So there are already several research papers have been published if you can have a look. So there are many papers wherein they have reported proteins and peptides from snake venom, which are showing good, inhibition of cancer cell lines. And yeah, so right now at this point, I can't specifically mention about those work and all. It's not in my head right now, but I can if you email me, definitely if you can just send me your email ID, I'll send you those papers. You can definitely have a look and you can plan your experiments. It has already been done,
Ma'am,
but of course, this field
shall
is
I
very
send it
vast.
in the chat or
Yes.
my email ID? Shall I send it in the chat or
Sorry, buying.
my mail ID, ma'am? Shall I send it in this meeting's chat or somewhere else?
Okay, okay, that is okay. Sure, you can send it in the chat. No problem. Yeah. Anyone else?
There is a chance for asking your questions and queries. If you have any, you can ask them now.
Ma'am, I have one question.
Yes.
Ma'am, in regards with metabolomics, any work has been done?
No, 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 not yet. Not yet.
Okay.
So I have just typed my mail ID in the chat box. So if anyone further you need any sort of information, any discussion, I'll be happy.